Hi everyone, this is lecture 19, Immune System. So today we're going to talk about the non-specific and adaptive immune responses. The non-specific immunity is also sometimes called innate immunity. So there are several components to innate immunity. We have external defenses, inflammation, interferon, natural killer cells, and the complement cascade. Then we have adaptive immunity, which is triggered by and orchestrated by the lymphocytes, the B cells and the T cells. So for adaptive immunity, we're going to talk about the B cell related antibody-mediated immunity. And for cell-mediated immunity, we're going to talk about the T cell related effects. So the immune system helps to guard against disease and tissue damage. Immune cells are distributed throughout the body. But as you just heard in our previous lecture, the blood lecture, they are highly concentrated in the blood. In addition to that, they will be highly concentrated in the lymphatic tissues. We haven't had much time yet to talk about the lymphatic tissues, so let's just remind you of those now. So the major lymphatic tissues will be the tonsils, the thymus, the lymph nodes, the spleen, the lymphatic vessels, which carry lymph fluid and also immune cells, Peyer's patches, which are mucosa-associated lymphatic tissue in the digestive system, the appendix, which also has lymphatic tissue, and the bone marrow. So the bone marrow is important for white blood cell and all other blood cell production. The function of the immune system is defense. It is to help the body against invading microorganisms. It is also cleanup. The immune system can remove damaged tissue and clean up debris. The last function of the immune system is surveillance. It can identify and destroy abnormal body cells. So again, the lymphoid tissues are going to be a primary site of the immune system. So unlike other systems, these tissues are not connected. So it's not like the heart connected to the blood vessels. These are all distributed throughout the body, so they are more connected by function and by cell type than they are by actual literal connections. So the bone marrow is the site of white blood cell production. The lymphatic vessels carry lymph from the tissues to the lymph nodes and the lymph organs. The lymph nodes, tonsils, and mucosa-associated lymphatic tissue, and the appendix will be large collections of white blood cells, these lymphocytes, for the immune response. They will also have white blood cells in the form of macrophages to remove debris. The spleen is important for replacing worn-out blood cells, but also has a collection of lymphocytes for the immune response. It will also have macrophages to remove debris. The thymus then is one of the sites of T cell maturation, and we'll talk about that in a moment. So to remind you, white blood cells, also called leukocytes, are less than 1% of the total blood volume. They are the major cells of the immune system. So wherever you find white blood cells increasing in numbers, you have immune system working. They are providing internal defense against foreign cells, removing debris, and destroying cancer cells. 5 types of white blood cells that we have already covered, but here they are. Neutrophils, lymphocytes, monocytes, basophils, and eosinophils. They can be distinguished histologically by appearance of both whether they are grainy looking, this would be the granulocytes when they're stained, versus non-grainy appearance, and also the particular morphology of their nuclei. The functions of the white blood cells, again to review, neutrophils are phagocytes. Lymphocytes are important for antibody production and adaptive immunity. We're going to go through all of that today. Monocytes are phagocytic. Eosinophils are related to allergic reactions and parasitic infection. Basophils are histamine allergic reactions and also can produce heparin. Okay, so let's talk about the targets of the immune system. What the immune system is looking for is something foreign. That foreign thing is called a pathogen. A pathogen is a disease-producing microorganism, for example, a bacteria or a virus. Bacteria is a living single-celled microorganism that can reproduce and grow on its own. For example, E. coli. So here's an example of E. coli bacteria. A virus is non-living but disease-causing, and it's basically DNA or RNA wrapped in a protein coat. I don't want to oversimplify viruses. They can be very complex in structure, and the only reason they're considered to be non-living is that they require host cells to reproduce. So they're very complex in structure, but they cannot live without a host. So they will take over the host cell and take over the machinery in order to reproduce themselves. There is a term called virulence, and that is the disease-causing power of any pathogen. So how strong or how effective are those bacteria and viruses at damaging the body, causing disease? So we're going to talk about several different aspects of the immune response, starting with the innate or nonspecific defenses. Then we'll move into the adaptive defenses to get a full multi-leveled approach to the immune system. So let's start with the innate responses. The innate or nonspecific responses are the first and second lines of defense to keep pathogens out of the body. So I think of this as, people say like, my body is a temple, right? Or my body is a castle. So you have sort of the external barriers to keep things out of the castle, keep things out of the body. If those fail, we can have some general troops that are protecting us against anything foreign, like our bouncers at the door. Okay. They should keep things out. If those fail and the pathogens get inside the body, then we will go to the adaptive defenses next. So let's start with these external barriers and more general cellular responses. First, the external defenses start with the skin. So the skin is a very large organ protecting the outside of the body. It prevents entry of pathogens into the body because it has multiple waterproof layers. So the keratinized epidermis is impermeable to most, not all, but most substances. So it's going to provide a protective outer barrier to the body. There are also immune cells within the skin called the Langerhans cells that can recognize pathogens that may enter the skin and alert the immune system. This is a reminder of skin anatomy, but I will not be testing you guys on skin anatomy here. If pathogens are able to get past the skin, they may have done it through the mucous membranes. So not only do we have the external defenses of the skin, but where we have openings to the eyes, to the mouth, to the nose, to other openings lower down in the body, vaginal openings, etc., we will have mucus. These mucous membranes are a major site of external defense. They're going to line these internal epithelial surfaces to remove debris and prevent pathogen growth. So we have to have openings in these regions of the body, and if we have to have these openings, they need to be protected. The digestive tract contains several protective components. Saliva contains antimicrobial enzymes, that's located in the mouth. It also contains mucus. Sticky mucus will trap particles. Stomach acid down in the digestive system will kill bacteria. The stomach is very, very acidic, not only to kill bacteria, but also to help with protein digestion. But it's handy that it's acidic because that kills bacteria. There's also good bacteria in the digestive system that helps to suppress the growth of harmful bacteria. In some of the lower openings of the urogenital tract, we will also have sticky mucus. Sticky mucus traps particles and prevents entry because of its stickiness. It will also be more acidic in these openings. So in the urinary openings and in vaginal openings, we will have more acidic secretions. So urine is acidic, which helps to flush out bacteria. Vaginal secretions are slightly acidic, which helps to prevent bacterial growth. In the respiratory tract, which is also open to the external environment, we have mucus throughout. This sticky mucus again traps the particles. We will also have hair and cilia. So the hair will be in the outer portion of the nasal cavity, and that will filter particles coming in through the nasal cavity. And then throughout the respiratory system, we will have tiny little cilia, sort of like microscopic little projections that will help to sweep debris out and away from the respiratory passages. If something gets down into the respiratory passages, way down into the air sacs, there are macrophages down in the alveoli that will help to clean up any debris. We also have a reflex in the respiratory system which can propel irritants out. That is the coughing and sneezing reflex that is so uncomfortable when you have a bacterial infection. So here's a picture of the cilia and the mucus trapping the particles, and then those tiny little cilia like hair-like projections that will be sweeping the debris out of the respiratory system. So those are external defenses: skin, mucous membranes. We have other innate or non-specific defenses that will just generally provide protection against bacterial growth. So inflammation is a non-specific response to tissue injury or foreign invasion. It involves phagocytic cells and plasma proteins that can isolate, clean up, and repair the area of injury. The important components of inflammation are macrophages and mast cells, clotting factors, chemotaxin molecules, monocytes, and neutrophils. So the stages and signs of inflammation are like this. You will have an injury. Macrophages will be first to the site of injury, and they will immediately come in and phagocytose or eat up any microbes and debris that they find. They can then secrete chemicals to alert the rest of the immune system. These chemicals will be chemotaxins. Chemotaxins means that they attract cells, and cytokines. The mast cells then will be recruited to the site and will release histamine. Histamine can cause vasodilation. Okay, think back to the cardiovascular system. What happens when you have local vasodilation? So you're going to go from a normal blood vessel to a dilated blood vessel at this local area of injury. The vasodilation actually increases blood flow to the injury. That will cause redness as the blood moves in and heat because the blood is warm. There's also, because of the histamine and other chemical release, increased capillary permeability. Increased permeability of the capillaries is so that more cells can come into the injury. So we're going to start to move white blood cells into the injury site. This process is called diapedesis, and that can cause swelling and pain, because not only do the cells move into the tissue, but fluid can move into the tissue. So increased fluid to the tissues can cause swelling. Then the swelling can push on the nerves nearby. That can cause pain. So the 4 signs of inflammation are redness, heat, swelling, and pain. After that, we can initiate the clotting cascade so the injury site can begin to be plugged off. The chemotaxins will continue to attract immune cells to enter the injury site, and the monocytes and neutrophils will move in to start engulfing and destroying pathogens. So again, these are white blood cells that will come in and start killing pathogens. As an aside, a bunch of dead white blood cells, because you had a ton recruited to the injury that accumulate at an injury, forms pus. That's a white exudant that is going to be pushing out of that injury, and that causes pus. When the white blood cells stick to the injury site, that is called margination. So let's look at these steps. I could draw them for you, but actually I think this picture is much better. So you have an injury that will cause an increase in white blood cells moving into the injury. That's diapedesis. The white blood cells will move in, neutrophils, mast cells, macrophages, monocytes. They're going to go in to try to clean up that debris. That is all caused by the increase in blood flow, which brings more white blood cells in, and the increase in capillary permeability, which allows the white blood cells to push through the capillaries. That increased blood flow can cause redness at the injury site. That increased permeability can cause swelling at the injury site. A result of the increased blood flow is also heat. A result of the swelling is also pain. So the signs of inflammation are redness, heat, swelling, and pain. We call these the cardinal signs of inflammation. These are caused ultimately by the vasodilation that brought in more blood flow to the injured tissue, and by the increased capillary permeability that brought in more fluid and cells to the tissue. That caused swelling and pain, which increased the phagocytes. Because of the increase in phagocytes in the tissue, you can also get an increase in pus at an injury site if it goes on for a long time. But more secretions can also activate the rest of the immune system, increase fever, and increase tissue repair. So here's a macrophage in action. So there's a bunch of E. coli, and there's a big macrophage kind of enveloping and bringing in the E. coli for phagocytosis. Now, phagocytosis looks like this. You have a foreign particle. This huge white blood cell is going to surround it and bring it inside. That is phagocytosis, bringing the particle in, and it will then fuse that phagocytic vesicle, which contains the pathogen and the outer membrane surrounded, with a lysosome. Remember that a lysosome is filled with digestive enzymes, so the digestive enzymes will break down and kill the bacteria. They will break it down into pieces and then exocytose those chunks of now non-damaging bacterial parts. This debris can build up and cause problems at the injury site if it goes on for too long. Okay, that was inflammation. Now let's talk about interferons. So interferons are antiviral proteins that help cells non-specifically target and prevent viral replication. So this is a specific antiviral defense. So we had external defenses, skin, and mucous membranes. We have a general response to pathogen entry, which is inflammation. And now we have a general response to viruses. So if the body detects viral nucleic acids, or it senses that there's a virus, then it's going to send out some autocrine and paracrine signals. This will trigger a virus-blocking enzyme in the body cells that will inhibit viral protein synthesis. This will also reinforce the immune responses generally by increasing phagocytic cells and activating other aspects of the immune system. It will ultimately slow cell division and growth in body cells as well. So interferon looks like this. You have a body cell that's been attacked by a virus. That first body cell is dead. There's no way to save that body cell. But before he dies, he sends out interferons. So this is sort of a, hey guys, I'm going down, don't go down with me signal. So he's going to release interferon, but he's toast, okay, because the virus has already replicated within him. That interferon, though, can be sent out to the next cell nearby. That interferon will then block infection of the nearby cell by viruses. So the nearby cell is now protected because his buddy told him to close his doors, right? Hey, there's a virus around, close your doors. This guy was toast, but he saved his friends on the way. So the virus then will try to get into the cell, but that cell has already been prepared and is able to block the virus from replicating. That virus will not be able to replicate, and that cell will be saved. So interferons are signals that are released from damaged, virally infected cells to help nearby cells prevent virus replication. Okay, natural killer cells. Natural killer cells are specialized to non-specifically detect and destroy virus-infected cells and cancerous cells. If they find a cell that has been compromised, either a cancerous cell or a virally infected cell, then the natural killer cells will release chemicals to kill those damaged cells. These chemicals are called perforins, and they're inserted in the membrane of the infected cell. Poking holes in a cell, not good, right? So the natural killer cell releases these perforins, it pokes holes in the damaged cells and causes those damaged cells to die. Complement system is similar, but it's through a larger cascade of molecules. There are 30 proteins involved in the complement system, but ultimately it is also going to poke holes in the membrane of a cell and kill those cells. So these holes are formed by what we call the membrane attack complex, not the perforin. So natural killer cells, perforins. Complement system, complement proteins, membrane attack complex. This forms a big channel in the membrane, and that disrupts the osmotic balance in the cell, bleb. So the cell is going to lyse and die. Complement's going to come up at the end of the lecture today because it also works together with other immune responses. Here's a zoom in of that membrane attack complex that pokes holes in those cell membranes. Okay, fever. Fever is caused by chemicals secreted by macrophages. So when macrophages find pathogens, or they find foreign invaders, they will secrete pyrogens. Pyrogens will be sent to the bloodstream and reach the hypothalamus. When the hypothalamus receives these pyrogens, it will elevate the body's temperature and cause a fever. A moderate fever can actually be beneficial. That's beneficial because a little bit of increase in temperature can increase the rate of chemical reactions. So think back to chemistry. The more you move a molecule with heat, the more active it can be, to a certain extent. That can actually speed up chemical reactions and enzymatic repair of tissues. It can also cause the liver and the spleen to sequester iron and zinc. Iron and zinc is needed by microorganisms to grow and divide, so that will help prevent microorganism growth. Excessive fever, however, I should have written that on here. Excessive fever can be harmful because what happens to enzymes when temperature goes too high? Right, enzymes are proteins, so the word I'm looking for is denature, right? So enzymes denature when proteins are high. Okay, so those were all non-specific responses. So let's list those out and do a summary diagram. Okay, so for the immune response, we have the innate or non-specific defenses. These are defenses that are general. They are generally going to be active with any foreign pathogen. Of course, there are some subclasses, so for example, interferons are mainly for viruses, but they will be active for any virus, not a specific virus. So they are general because they are grouped and they will respond with any infection. First, we have the external defenses, which are the skin and the mucous membranes. The skin and the mucous membranes are going to prevent entry of pathogens like bacteria. Then we have inflammation, which is generally going to try to increase blood flow, white blood cells, to an injury site. As a byproduct of that, we will get the 4 cardinal signs of inflammation: redness, heat, swelling, and pain. But ultimately, inflammation is a natural response to injury. The last 3, interferons, natural killer cells, and complement, are going to be targeting specifically infected cells. So interferons are going to be antiviral. Natural killer cells are going to use perforins to destroy damaged cells or infected cells. And then finally, complement is going to use the membrane attack complex to destroy damaged or infected cells. All right, let's move on now to the adaptive responses. The adaptive responses are the 3rd level of response. So if the external barriers and the internal general defenses don't work, so if inflammation is not enough, if the natural killer cells and complement and interferon are not enough, then we need to do more. So we need to then target specific pathogens that have entered the body. And this is where the adaptive immune response comes in. The adaptive immune response creates a memory for those very difficult specific pathogens. They will then be recognized upon later infection, and they will be killed more efficiently. So the adaptive responses are 2 different types. Antibody-mediated immunity, antibody-mediated immunity, which happens through B cells, and cell-mediated immunity, which happens through T cells. So for adaptive versus non-specific immunity, the adaptive immune system is specific. And we say that it's specific to a particular foreign invader. In fact, it's specific to a piece of a foreign invader called an antigen. So this requires the production of specific lymphocytes, B cells and T cells, and specific antibodies against an antigen. It is systemic, so it's not restricted to the initial infection site the way that inflammation is, and it has memory. A second encounter with a pathogen that makes this tiny or has the antigen as its component causes a more rapid and vigorous response. So this is driven by the B cells and the T cells. The B cells are named B cells because they are born and mature in the bone marrow. They are then housed in the lymphoid tissues. B cells are going to recognize free pathogens, bacteria, toxins, viruses floating freely in the body. They are going to produce and create the antibody-mediated immune response. T cells are born in the bone marrow, but they mature in the thymus. So they get their T for where they mature in the thymus. They are housed in the lymphoid tissues, just like B cells. They can recognize infected or cancerous cells. So the B cells are going to recognize the whole pathogen. The T cells are going to look for body cells that are infected. They will be involved in cell-mediated immunity. They are also involved in activating a total and combined immune response. So in order to do this, B and T cells have to become what's called immunocompetent. That means that they display a particular type of receptor that responds to a particular type of antigen. I'm realizing that we're using the word antigen, but we haven't completely defined it yet. So let me show you briefly what an antigen is. So what you have is a bacteria. This bacteria, as a whole, is considered a pathogen because it's a disease-causing microorganism. They are very complex and they contain many different molecules around them, particularly in their outer walls. When a macrophage finds a bacteria, it's going to take in the bacteria through the process of phagocytosis. When it does that and fuses with the lysosome, it's going to break down the bacteria. When it breaks down the bacteria will now be in pieces. Those pieces will include molecules that originally made up the bacteria. Those pieces are antigens. So an antigen is a molecule that causes an immune response. And it can come from pieces of pathogens that have been broken down in this process. It can also come from pathogens that are just floating around in the body that have previously caused immune response. But an antigen is basically a molecule. It's not a complete pathogen. It's just a portion of a pathogen. And the important piece here is that one pathogen can have many antigens. One pathogen broken down into many molecules, those molecules specific to the pathogen will be the antigen. So to become immunocompetent, the B and T cells need to have receptors that will find these antigens. So these are receptors that are specific for that molecule. They need to become immunocompetent before they encounter the antigens so they can actually recognize those antigens. Once they do that, they will be exported to lymphoid tissues where the antigens will be encountered. Once they actually encounter their antigen, they will become fully functional and be activated. So for B cells and T cells, the pathway is slightly different. They're both made in the bone marrow. B cells will stay in the bone marrow and get their receptors or become immunocompetent in the bone marrow. T cells will leave the bone marrow and obtain their receptors and become immunocompetent in the thymus. They will then both circulate to the peripheral lymphatic or lymphoid tissues. Here's the antigen definition. Antigens are any molecule or partial molecule that triggers an immune response. These can be cell proteins, carbohydrates, lipids that are part of bacterial cell structure. They can also be toxins released by bacteria. B and T cells will then have specific receptors for specific antigen molecules. B cell receptors, we're going to abbreviate BCRs. T cell receptors will abbreviate TCRs. We could spend an entire class on immunocompetence. It's a very complicated series of interactions that ensures that B and T cells have receptors for foreign antigens and not molecules in our own bodies. There are 2 trillion lymphocytes in the body, and each lymphocyte can only recognize 1 out of 100 million possible types of antigen. Okay, B cells. So we're going to first talk about antibody-mediated immunity and what the B cells do. So here's what happens. The pathogen invades the body, and the specific B cell, with its specific receptor, because it's now immunocompetent, recognizes a specific pathogen. The B cell will then differentiate into plasma cells and memory cells. Plasma cells produce antibodies. Memory cells store a memory of that antigen so that if you have a later infection, weeks, months, days, years later, then they will be ready to produce antibodies quicker the next time. So this is how your book diagrams it, but we're going to draw it out. So you have a bacteria that has invaded the body. This is a free floating pathogen circulating somewhere in the body tissues. That bacteria is going to have on it some molecules that will be specifically recognized by our B cells. So we have B cells with specific receptors for those specific portions of the pathogen. So these are antigen receptors. And let's just follow this pink B cell. So this particular B cell has a specific receptor for this molecule on this bacteria. It's going to go near the bacteria and recognize that molecule. When it recognizes that molecule, the B cell is going to be activated. In other words, it found its soulmate. It found its specific antigen. Right? So they could go their entire lifetime without ever encountering the antigen that they have been built to encounter. But once it does, it will become activated. So this is a B cell with its B cell receptor. Once it becomes activated, 2 things happen. The first thing that happens is it produces plasma cells. Plasma cells are going to produce antibodies. The second thing that's going to happen is it's going to produce memory cells. Memory cells are going to remember that antigen later. Okay. So instead of having just one B cell or one small group of B cells that recognize this antigen, we're going to now have a collection of memory cells that can recognize this antigen upon a later infection. So what is the significance of these antibodies that are produced by the plasma cells? So here's your bacteria, here's your B cell with a specific receptor for the antigen on that bacteria. Other B cells with other receptors will not be activated. That specific B cell will become activated and make plasma cells. Plasma cells will make antibodies, and the antibodies are also called immunoglobulins. They are Y-shaped proteins that recognize specific antigens. There are 5 types of antibodies that are created in the body. The most common is IgG. IgG will be secreted to respond to most antigens. So for example, you're infected with disease bacteria X, I totally made this up, DBX. You will have a specific type of B cell that specifically recognizes DBX. That will activate plasma B cells to produce the IgG specifically for a cellular protein on DBX. And then what happens? This is the structure of an antibody, so you have to wait for what happens for me to show you the structure. Sorry, I forgot this slide was in here. So antibodies are Y-shaped proteins, and they recognize specifically the antigen. Okay, so then what happens? We have the antibody-mediated responses. It's important to know that the antibodies do not kill the bacteria. The antibodies cause a couple of things that can prevent the bacteria from doing more harm or can downstream lead to the destruction of the bacteria. These 3 major processes are neutralization, agglutination, and tagging for destruction. So antibodies can physically surround a pathogen to prevent it from interacting with other cells. So it can form a barrier around that pathogen. So it will bind to the outside of that bacteria, for example, and prevent it from interacting with cells. It can also clump molecules. So it can clump bacterial cells together. So antibody will grab onto one cell, another to another cell, they'll all clump together, and it can cause them to precipitate out of solution. The last is that antibodies will tag a cell for destruction by something else. So the antibody itself doesn't do the destroying. The antibody will tag it and say, hey, I found something, and then the complement, the phagocytes, and the natural killer cells can come in and either gobble up the bacteria or poke holes in it so that it dies. Here's agglutination. So this is antibodies clumping together several bacterial molecules. So here in brown, these are the bacteria. Here in red is the particular portion of the bacteria, the antigen, and then here's the antibody clumping all of these together. Here's the activation of complement. So the antibody the antibody binding to the antigen will lead to active complement. And then through the cascade, complement will form the membrane attack complex around that bacterial that was found and cause lysis or rupture of that bacterial cell. So the complement does the killing, the antibody is just the flag. Activation of phagocytes happens in a similar way, where they will be attracted to the antibody bacterial complex, and they will surround the bacteria and phagocytose the bacteria. Natural killer cells, very similar, but they use perforins instead of the membrane attack complex. So how long does this take? Antibody response time depends on whether it's your first exposure or a later exposure. The first exposure to an antigen is very slow. It can take several weeks before antibodies can reach their peak concentration and the bacteria or other pathogen invasion can be held back. In the meantime, symptoms can get worse, and sometimes death can occur before the antigen has been neutralized. The secondary response is the second or later exposure to the antigen where you now have memory cells, and the memory cells can respond very quickly and much more powerfully. So memory cells can make enough antibodies within just a few days. The secondary response to that antigen may not even create symptoms, so you might not even know that you got sick the second time, and later exposures after this primary response will be completely contained. So this is the basis of vaccination. So the idea of vaccination is to mimic the primary response. So what you do with vaccination is you take a piece of a pathogen, something that cannot cause disease. Remember that an antigen is just a molecule. It's a chunk of that bacteria or a piece of that virus. It cannot fully cause the disease, but it can cause an immune response. The non-pathogenic vaccine then will be the primary exposure to that antigen. It might take weeks after that vaccine to develop immunity, but after that, you will have memory cells. And because that portion of the bacteria or virus cannot make you sick, then you created memory cells without getting sick. So you skip the primary infection and go right to having memory cells, and you're then prepared for any other exposures. So this will be done for diseases in which the primary response could create enough damage or could be fatal. In that case, it's very important to get a vaccine to prevent that initial infection and to guard against any exposure to the antigen in the future. So this is the time of exposure to a first antigen, and it takes a couple of weeks for that to get enough antibodies to make a difference. This is the second exposure, and you can see within just a couple of days, so before the first week is over, you have a much larger response because of the memory cells. Let's put these two together then. So here you have the first exposure to an antigen, which can take a couple of weeks to create enough antibodies. We will then guard against that pathogen, and the antibodies will go back down. The pathogen will be destroyed. Then if you get a second infection, it will take only a couple of days to mount a much bigger and more powerful response and make enough antibodies to ward off the infection. This is what we call active immunity. B cells encounter antigens to produce antibodies against them. This can be natural in response to a regular bacteria or viral infection, or it can be artificial in the case of a vaccine. But either way, the patient is exposed to a bacteria or virus molecule or a portion of that bacteria or virus that is either natural or artificially put into the body, and that will cause memory cells to help against infection later. Passive immunity is different. Passive immunity is when you give a patient antibodies. So in this case, the cells are not challenged by the antigens, B cells are not activated, so you don't get memory But you can protect someone by artificially giving them a bunch of antibodies that will help to neutralize, agglutinate, or tag the bacteria for destruction. So these can be natural. This happens with breast milk from mother to baby. There are antibodies that are passed on to the baby. Or it can be artificial, where a clinician will inject a serum that contains antibodies. In both of these cases, though, because the B cells are not challenged and do not bind to antigens, we skip that step and just go straight to the antibodies. There's no memory. So this doesn't help with long-term immunity. It only helps while the antibodies are being administered. Okay, so that was B cell immunity. We're going to finish up now with T cell related immunity. So T cells are going to search for infected body cells. So B cells are searching for bacteria. T cells are searching for damaged or infected body cells. If the pathogen has been successfully able to get into a body cell, then that body cell is going to send out signals. This requires both the recognition of self cells and infected body cells. And this is done through MHC proteins. Okay, so MHC proteins are on the surface of cells, and they can signify a self cell. They are also used on antigen-presenting cells, and we'll talk about those in just a second. But the idea here is to coordinate and enhance other immune responses. So there are 3 types of cells used in the T cell response. There are cytotoxic T cells, helper T cells, and regulatory T cells. So let's talk about the antigen-presenting cells. So what happens is that T cells cannot recognize free pathogens the way that B cells can. They can only recognize antigens displayed by MHC proteins. So there are 2 possibilities. One is that a pathogen infects a body cell. The pathogen will be broken down inside that body cell in the lysosome, and an MHC1 protein will be used to display the pathogen on the cell surface. This is a body cell saying, I have been infected, and this is what I'm infected with. MHC proteins are also used on cells we call antigen-presenting cells. These are specialized macrophages that will phagocytose pathogens or bacteria, break them down in their lysosomes, and then display them on an MHC2 protein. We now call those macrophages, which the name for those are dendritic cells, we now call them antigen-presenting cells. So they are hovering around the body looking for pathogens. When they find a pathogen, they will grab it, break it down, and use an MHC2 to display it and say, hey, I found something, here it is. So here is a dendritic cell engulfing a bacteria. This looks very much like the same phagocytosis process we talked about. So the dendritic cell engulfs the bacteria, brings it inside, breaks it down with lysosomes, and then pieces of that bacteria, these antigens, will be displayed. And they're displayed by MHC2 molecules. Now, the MHC2 molecules will be recognized by the T cells. So those 2 general classes of MHC found in the body. Class 1, MHC, signals a self-body cell that is infected. Class 2, MHC, signals an antigen-presenting cell that is flagging a pathogen has been found. Okay, so let's begin to draw this out because it's getting complicated, yes? Okay. Okay. You have a bacteria. That bacteria is engulfed by a dendritic cell. It is the dendritic cell's job to find foreign invaders. And it found one, so it brings it in and breaks it down. It breaks it down and then uses an MHC protein to display it. So this is an antigen being displayed by a dendritic cell on an MHC protein. When the dendritic cell is displaying an antigen, it is now called an antigen presenting cell. Once the antigen presenting cell is displaying an antigen, it's going to be recognized by a helper T cell. This is one of these T cells that is involved in the response to these displayed antigens. So the helper T cell has its own T cell receptor. So this is our helper T cell. It's going to be floating around and looking for antigens on antigen presenting cells. When a helper T cell finds an active antigen presenting cell, it's going to release signals that will activate the rest of the immune system. They're going to activate B cells so that the B cells can look around for the same bacteria floating around and make antibodies. And they're also going to activate something called cytotoxic T cells. The cytotoxic T cells are going to then take that information and start to look in the body for infected cells. So the cytotoxic T cells, let's draw them in pink actually. So now the cytotoxic T cells, armed with information that there's an antigen to be found, are going to look for that same antigen that was found by the helper T cell. When they find that antigen, they will recognize it by binding to an antigen that was on an MHC protein. MHC will be attached to an infected body cell. So this is a cell that has been infected and has displayed the cause of its infection, the antigen on its surface. So the cytotoxic T cells as activated by the helper T cells start to circulate and look for infected body cells. Once they find infected body cells, they're going to destroy those body cells. Those body cells have been infected, so they're on their way to being damaged by the bacteria. We want to make sure that they don't create any more damage. So the cytotoxic T cells are going to cause lysis of these infected body cells. We've already heard about two ways that lysis can be induced. Perforins from natural killer cells, the membrane attack complex from complement. Now here's one last way, granzymes. So the cytotoxic T cells release granzymes to induce lysis of infected body cells. Now these can be virus-infected cells, they can be mutated unhealthy cancer cells, they can be transplanted donor cells. In any case, if the cytotoxic T cell finds an antigen on an MHC it's going to kill that body cell. This was helped by the activation of helper T cells. So helper T cells activate B cells, they activate these cytotoxic T cells. They will also generally attract macrophages. That includes neutrophils to help to find more antigens and more debris. There are also some cells called regulatory T cells. I don't have an image for these. They are currently under research. But regulatory T cells are thought to keep the immune system in check. So they are thought to reduce immune responses, and they are currently being researched for autoimmune disease. So when the self versus pathogen signals get mixed up, then the body can mistakenly attack its own healthy cells. Regulatory T cells can keep that immune response lower and hopefully prevent autoimmune disease. Okay, there are some summary diagrams in your textbook for this, but let's take a moment to put them back together for B cells and T cells. So here's our B cell response. B cells recognized free-floating pathogens with their specific antigen receptors. When they find those free-floating pathogens, they will activate production of plasma cells and form antibodies. They will also form memory cells that will remember the antigen later. T cells are here. They are going to recognize antigens only as displayed on MHC proteins. So T cells will first be activated by antigen-presenting cells. Antigen-presenting cells will activate helper T cells, which will overall enhance the immune response in the body. That will also increase B cells and macrophages, but primarily for the T cell response, it's going to increase cytotoxic T cells. Those cytotoxic T cells then, based on MHC1 interactions, will kill any infected body cells. So T cells kill infected cells. That's why the T cell related is called cell-mediated immunity. And B cells are going to kill bacteria with antibodies, so that's why the B cell is antibody-mediated immunity. You have a summary diagram in your textbook which talks about the differences between B and T cells. One example of antibody use clinically is individual blood type. So individuals have particular proteins expressed on their blood which mark it as self versus non-self. If you are blood type O, you don't have any of these proteins. If you are blood type A, you have A antigens. If you're blood type B, you have B antigens. If you're blood type AB, you have A and B antigens. If you are an A containing blood cell, then your body will only recognize A as self. If it encounters anything else, namely B, it's going to make anti-B antibodies. The same goes for B-type blood. If you are a B-type blood, then your body recognizes B as self, and if it encounters anything else, it will make antibodies to that, namely anti-A antibodies. If you don't have any cell markers, then both A and B will be recognized as foreign, and you will make anti-A and anti-B antibodies. In any of these cases, whatever is recognized as non-self will be attacked by the immune system. That can cluster and cause clumping of donor blood if you give a patient the wrong blood type. And that's the agglutination response that we see normally with antibodies applied here to blood type. Hypersensitivity is an abnormal or extremely vigorous immune response that can occur within seconds to minutes. This can occur with allergies and with asthma. So this can happen when plasma cells produce IgE antibodies, which attach to mast cells. Mast cells then release histamine, causing a huge inflammatory response. They can also release a leukotriene that causes a slow substance that causes smooth muscle contraction in the airways. That will constrict the airways and cut off airflow. This can be treated, though, with antihistamines and newer drugs that block leukotrienes, such as Singulair. Immunodeficiency is the opposite. It's when you don't have enough of an immune response. Immunodeficiency can be caused by lacking or abnormal production of immune cells. So you can have something called severe combined immunodeficiency disease, which is deficiency of both B and T cells. And then out in the media discussed a lot is AIDS. So acquired immune deficiency syndrome happens because HIV specifically destroys helper T cells. Helper T cells are necessary for activating cytotoxic T cells to kill infected body cells. They're necessary for activating B cells. So the entire immune response will be lowered by the destruction of helper T cells. So you don't die from AIDS, you die from a secondary infection that you weren't able to fight because your T cells were destroyed. An autoimmune disease happens when the immune system does not distinguish well between self and non-self. In this case, something on a self cell will become an antigen, and the body will produce antibodies and cytotoxic T cells that are going to attack its own tissues. There are many possible causes for autoimmune diseases, but the research here still is very ongoing. Some of this could be inefficient lymphocyte programming, for example, a problem with the immunocompetence development. This could also be the appearance of new self-antigens, something new that's created in the body. Or it could be that the antibodies produced against a foreign antigen could begin to react with a similar self-antigen. Okay, we are finished with the immune system. Please take a moment to review the nonspecific defenses, the B-cell mediated antibody response, and the T-cell cellular mediated response. Let me know if you guys have any questions.